Hello, I'm Munira, and today's topic is genetics. We'll be defining genetics. We'll be mostly focusing around the terminology in genetics and inheritance. We'll also look at Gregor Mendel's concepts around segregation and independent assortment. And we'll also do a quick monohybrid cross. Enjoy! Did you ever notice that you could look similar to your parents and different to your siblings? Well, this is genetics. Genetics is the study of heredity. That means that something is being passed down from your parents, another generation, to you, the offspring, or next generation, and the study of variation. Variation, as you can see in the offspring, they all look different. Some are daughters, some are sons, some have blue eyes, and some have brown eyes. Let's talk about the characteristics that you find within humans. We'll mostly focus on humans. So some of the characteristics is a hitchhiker's thumb. Now, I think my one thumb is a hitchhiker's thumb, but the other one, not so much. Because if you look at this side, one's really straight up and one's really curved. From this picture, you can see the hitchhiker's thumb is really curved out, and the no hitchhiker's thumb is quite straight. Then we have attached earlobes and non-attached earlobes. I think I have a lobe over here, so I have unattached earlobes. Okay. Then the other characteristic is to roll your tongue like that. Can you roll your tongue? Okay. All these characteristics make you you, but where do they come from? And how do we know where they come from? So how is genetics studied? Well, they look at cells. This is an animal cell. Within the animal cell, we find a structure that controls the cell's functions. This is called the nucleus. Within the nucleus, we find a chromatin network. The chromatin network is interconnected strands that consist of material that codes for life. This is called DNA. We know if we take DNA out that it is a double strand with different nucleotides. If we take a segment of the DNA, it is called a gene. Now we know the DNA has thymine, guanine, adenine, and cytosine. Now what makes a gene is that it has two or more triplet bases. A triplet base is a group of 3 nucleotides. So there's one and there's one. When a cell wants to grow or multiply, the chromatin network has to coil up to form chromosomes. Let's look at this example. Let's say this is our chromatin network. When you want to move out your house, you don't just move freely. If you want to grow, you have to pack your luggage. This is what the DNA does. So it coils up and forms chromosomes. Similar size chromosomes will link up. Okay. Now if we look at the DNA strand, we can see little segments. These can represent genes. No, not the genes that you wear. The genes that code for a specific trait, like hair color, eye color, hair texture, size of your nose, things like that. Now, why does 2 chromosomes pair up, and why do they have to be the same size? Well, from meiosis, we know that homologous pairs of chromosomes line. Well, one is from the mother and one is from the father. Why do they have to be homologous? Well, homo, the prefix, suggests that they should be the same size and shape to exchange material. Where do we find genes on this chromosome? Well, we just take a segment of the DNA. This is the gene. This could code for eye color. Okay. With the other chromosome, there could be the same gene that also codes for eye color. Alleles is an alternative form of the same gene on a chromosome. So this one codes for eye color. This one also codes for eye color. So that is an allele. It is also the same. Then we get the location where the gene is found. This is called the locus. So who is the person that discovered there are 2 chromosomes that align that are homologous pairs? Well, his name is Gregor Mendel. He is known as the father of genetics, and that he discovered hereditary characteristics are determined by 2 genes. So one comes from the mother and one comes from the father. He also has 2 laws. His first law states that the pair of genes are able to separate and form gametes on their own. Okay? And that is his law of segregation. His second law of independent assortment means that one characteristic does not influence the other characteristic. Okay? Genes for different traits assort independently. This means that if you are tall and you have blue eyes, doesn't mean that you can't be short with blue eyes. And if you have brown eyes and you're tall, doesn't mean you can't be short and have brown eyes. Similarly, you can be tall with blue eyes and be tall with brown eyes. These traits assort independently. The segregation law helps us when we do crossing over, which we'll do soon. There are always 2 genes that represent a characteristic. These are represented by letters. Over here, we are looking at different lip sizes, the thickness of the lips, and thinness. So it is represented by a capital T and a small t. How do these traits play out? Well, they have a genotype. Now, genotypes come from the word gene. Yes, that means inside. What 2 genes do you have that represent your characteristic? Okay, are they identical? Which means, are your 2 genes the same? Are they both coding for the same characteristic, or are they non-identical? Are they coding for different things? If they are identical, we call them homozygous. Okay? And if they are non-identical, we call it heterozygous. Okay? Homo means the same and hetero means different. And zygous means the origin. So does it have the same origin or does it have different origins? Okay. Now the phenotype, which as you can hear with phenotype means the physical features. Okay. The phenotype does not speak about identical or non-identical, homozygous or heterozygous. The phenotype is what is expressed, what you can see. Here we can see thick lips and thin lips. This one has a small t and a capital T. This is heterozygous. They are different. So genotype is heterozygous. However, it has thick lips as we can see in its phenotype. This brings us to what is dominant and what is recessive traits. Dominant traits are represented by capital letters. Recessive traits are represented by small letters. But what is dominant and what is recessive? Well, if you hear the word dominant, you can hear, oh, it's the stronger, it dominates. If something dominates some other thing, the one is stronger than the other. So when we see a dominant trait, we will not see the recessive. Now the saying goes, the mouse only comes out when the cat is gone. The cat is much stronger than the mouse, but the mouse will only be seen if there is no dominant trait. Why do we see thick lips in this person? Because there is a dominant trait here. So now we'll be looking at the mono-hybrid cross. Mono means one, which means we'll be only looking at one characteristic. So let's choose one person has blue eyes and one person has brown eyes. Now in our case, we'll choose blue eyes to be recessive and brown eyes to be dominant. Okay, eyes are dominant. So first we start off with our P1, which means our first generation. We'll look at the phenotype, blue and brown. Then the genotype. Remember, genotype needs to have 2 genes. Okay? So blue-eyed has 2 small R's and brown eyes has big R's. Okay? Then they go through a process called meiosis. Through meiosis, they form gametes. And then they go through fertilization. Okay, that's the processes. To the genes, when they form gametes, they separate. So 1 R, 1 R, 1 R, 1 R. Okay? And then they'll fertilize and form a F1 generation, which is the offspring. Now how do we calculate what genotype and phenotype the offspring will have? This is why we form a Punnett square. We write gametes in the middle, at the top, and we have our big R, and our small r, small r. Now we do multiplication. The big R, small r, big R, small r, big R, small r, and big R, small r. The genotype of the offspring will be 100% big R, small r, and there will be 100%. What is the dominant trait? Brown eyes. Let's do a quick recap on the whole topic. What is genetics? Genetics is the study of heredity and variation. what did you get from your parents and what is different from you to everyone else. That is genetics. Then some of the terminology, we know that within the cell we find DNA, and if we take a strand of DNA, only a segment, how do we study genetics? We study it through genes. Look at our characteristics. They need to be determined by two genes. One comes from the mother and one comes from the father. Your phenotype. Your phenotype is the only thing that is expressed. You won't know if you have a dominant or recessive trait on your two chromosomes in your phenotype. You will only know that in your genotype. How do you work that out? Well, if you look at the previous generations before you or generations after you. That is why we did the monohybrid crossing. Please let me know if this helped you with your genetic studies, and I hope you enjoyed the video. Bye!